going old school then. Okay. I'm not going to sing today, I promise, you don't know. Thank you for having me, and my name is Zofia. As I was introduced, I'm from EditCo Bio. We are one of the partners. We are very happy to be here today, and I'm going to talk today about how the cell models actually change the research, and why is it important to think earlier when it comes to appropriate cell models in drug discovery space. But before I start, I wanted to ask, how many of you actually heard of EditCo before? Can I have them hands up? Not so many. We talked before already, but this is good. That makes me actually very happy that I can introduce you to some new technologies out there, or a new company. We have been on the market actually for more than 10 years now when it comes to cell development using CRISPR. We came from Synthego, so you may have heard of Synthego before, so we essentially spun out from them last year. I know it's right before the break, so I wanted to keep it interactive, and I have a little game. So if you can find, without this one, the smiley DNAs, you can win a coaster that says the genius at work. So feel free to go ahead and talk to me after and tell me how many you found. Hopefully it will keep you awake as well until the break. But we are working all in the drug discovery space, and I wanted to say why is it important in general when it comes to cell models, why they're important in that space. We all talked about how we need to fail faster, and one of the main reasons, which was in this paper from Nature Reviews, is that the lack of efficacy is actually the key component why we fail so late in the drug development space. So we need to fail faster, we need to have better cell models to get to where we need to get to when it comes to basically clinical trials and launching drugs on the market. But where does it all come from? Well, actually, here we talk about the GWAS, but we talked a lot about the AI. We have lots of places that we can find the potential targets, disease linkage with the point mutations and things like that. But what do we normally do? And I am also guilty of that. I was a bench scientist myself. What I usually do, or used to do, I was using the common way of filtering whatever I could find to develop the cell models in our lab, essentially. And then, for some reason, we limit ourselves to around 5, maybe 7 cell models that we are studying. So in this case, maybe point mutations, 5 point mutations or 5 knockouts if you want to do the target validation. And then with the attrition rate that we have in the drug discovery, we essentially launched 0.01 drug on the market, which is definitely now where we need to be. We need to switch it. So how we do that? We need to think bigger. So let's imagine doing the comprehensive screening. And I know some of you already talked about it, that we do the whole human genome screening and things like that. So that's definitely something possible. But let's think about doing this comprehensive screening, analyzing hundreds of point mutations, hundreds of cell models that we study, and then essentially launch the drug on the market. So it's not about even failing faster, it's also about increasing the chance or even getting into the market in a successful way. So it wouldn't be just imagination if I wasn't here. I don't want to leave you hanging and thinking that this is not something possible. So that's where EditCo comes in. And we can support you with the do-it-yourself reagents to do CRISPR. That's mostly the knockouts. And we can also take the whole work off of your hands. So you can come to us and say, I want to do the immortalized cells, iPSCs, or primary cells. I want to do knockouts or knock-ins. Whatever it is, we can essentially do the cell model customized to your needs. And we've been working very successfully with lots of biotech and pharma already on the market. But when we look at this slide, actually I left the brochure on each of the tables, so you can come back to it if you need to later on. But when you look at it, lots of it is related to the knockouts, higher throughput screening, thinking bigger when it comes to the actual knockouts. But then that's why I want to talk to you a little bit about the strategy that we actually use for the knockouts, because I think that's in our technology. So we use up to 3 good guides working in one early exon, and they cause the fragment deletion, and that fragment deletion increases the editing efficiency to the level that we can actually get up to 100% editing efficiency at the pull stage already, which is obviously very high. But what it also enables you to do, I know that there are lots of different screening technologies when it comes to CRISPR, but it allows you to do one PCR reaction to do your NGS or Sanger sequencing to validate your data. This is not something that is possible with any other technology on the market, and it's proprietary to EditCo Bio. And everything is already pre-designed. So if you work with one gene, or if you do whole human genome screening, you have a full money-back guarantee essentially that you will get the data that you should be getting. So you will be getting the successful knockouts. So as I mentioned, if you wanted to do it yourself or any of your scientists wanted to do it themselves, then definitely you can use the gene knockout kit. It's work one gene at a time. And we actually have a promotion for the new users. You can reach out to me and I can give you the code. It's also listed over here. So if you wanted to give it a try, you can literally put this code into the ordering page and it will get you the 50% discount. If you work at the higher scale, from 20 genes onwards, that's when the RA CRISPR screening come into play, and that's where essentially you can customize your library completely for your needs. And that's how our lab looks like. There's a compound on the right that you can see that has all of the multi-guide for the whole human genome pre-designed, and then the robot on the left is plating everything according to your needs. If you wanted to do like a heart shape, basically wells in your RA screening, you can. It's completely up to you. You can choose any of the pathways that we use, or you can give us your own pathway that you want to plate on the plate as well. But then usually the main question is like, what if I don't want, or what if I don't want to do it myself, or what if I don't even have a capacity to do CRISPR myself? And this is actually a very, very fair question. Because we did a benchmark study back in our Syntical time actually, but I think it's still very valid. So if any of you did CRISPR before, you will know that probably some of those numbers resonate with you. So 43 is an average editing efficiency that also experienced scientists get when it comes to doing the CRISPR. That's knockouts and knock-ins, but still not very high editing efficiency. Second is 7 is number of attempts that actually takes you to get to this 43% of editing efficiency. And 100, which is a big one here, is number of hours that actually takes you to get to the successful clone. Now, if you want to work at a higher scale, I don't think anybody has that much time to actually do it themselves, if you wanted to really do the full perturbation. So that's when we come in, and I hope that what you can see before the future sign is something that will excite you as much as it excites me whenever I see it in real life as well. This is our lab, which is in California, very close to San Francisco area. If you're ever around, let me know. I'll be happy to organize a tour for you with our scientists. But I think that really shows you when you can industrialize developing the cell models for your needs. So what we did in the past, we were testing any cell line that was getting into us, and we were doing 200 optimization stages. You remember from my previous slide, 7 is average that the customers or our researchers do it basically in their lab. So we were doing 200 optimizations, and now we've optimized over 1,400 lines on our platforms, including primary cells, iPSCs, and immortalized cells. And it's very important to see the difference in editing efficiency or ease of transfections when it comes to different cell types. So here we have the immortalized cell type, but things like THP1s, I don't know, anybody ever tried to edit THP1s in here? Like, I would, oh yeah? How was the editing efficiency in your hand? Bad. Yeah, so like what I hear actually the 7-10% is roughly what people get, even experienced people, right? Like that's what they tend to see. We get to 82% for the knockouts already at the pool stage. So sometimes you don't even need to get to clones, spend all of the money on the clones. You can do the preliminary study with the pools, which are cheaper, and then they can be delivered in as little as 4 weeks to you. And everything is custom, you are the owner of the cells, you're good to go. What happened? Okay, sorry. It just jumped back for some reason to the other slide. But yeah, I talked a lot about the knockouts, which are in heart on our technology. But there is a big chunk actually of the things that we can do, and maybe this slide and overview doesn't show that. And actually, I was talking to my colleague Hasti yesterday that literally I realized that yesterday that we didn't give too much justice maybe to that part of the portfolio, which is the knock-ins. So when it comes to knock-ins, there are lots of different things that we can do. That would be anything like point mutation, so doing the disease linkage between the healthy donors and the patient. So if you want to reproduce the mutations, we can do that. We can do things like HiBiT, luminescent fluorescent tags for you to track the protein of choice. We can use the degradation tags. We can do similar things to PROTAGs essentially as well. Affinity tags, as well as going all the way to the reporter and fusion genes, right? So something from very small to the very big knock-ins, but anything is possible. Just reach out and we can talk about it depending on your assay. But what I wanted to talk to you about today is something that we are very happy to announce as quite news. I think it's been 3 months at the moment, that we have started a partnership with a company called Promiga. You probably heard of them before. We are their official partner to do the cell model development for them. So even if you reach out to Promiga to develop a HiBiT line, they will come to us to produce it. But essentially, if you haven't heard of HiBiT before, this is something that you can use instead of, for example, using the antibodies for the detection. So it has very high luminescence signal when it combines with the large bit. And essentially you can detect and measure the protein in real time. You can detect receptor internalization, quantify the protein abundance and response to degradation, as well as track the protein changes in response to treatment. So whenever you do later on like small molecules, for example, and you want to see which one is affecting your protein the most, this could be also a good level because luminescence will show that. And HiBiT is very powerful by itself, but you can actually combine it also with the degradation tags. So that's a very good and very popular technology used recently by our customers. So we can tag any protein of your choice with dTag and the HiBiT, and essentially you will see, like, this is a slide from the papers, which is not our paper, it's just like for the research purpose really here, just to show you that essentially on the left you can see the response to the different levels of the dTag, so the compound that you're essentially adding to your cell culture media, and on the right you can see the response in time. And then on top we can see the level of the luminescence and the drop of the luminescence based on the protein degradation. That's based on the HiBiT tag that was added to the degradation tag. And if you don't know how to measure luminescence or you don't have the tools to do that, then you can also get an antibody against the HiBiT, and you can do it also with the Western blot. So this is a really great tool if you want to use even knockout alternative when you study the proteins or the genes which are essential to the cells. You could use that kind of technology to tag them. And also for all of the other things listed on the right hand side, so feel free to reach out if you are having struggles with the classic knockouts, or if you wanted to have more in-depth information whenever you study and whenever you use the cells in your assay. So what you get when you work with us in general, I think that comes back to 4 major things. We have a very precise and reproducible edits, very high editing efficiency both for knockouts and for knock-ins. They are delivered very fast. The pools take around 4 weeks. The clones, of course, we grow from the single cell. So we need to wait until the cell grows. So you do the single cell printing and then grow the clones from those. You get 2 clones and 2 vials each if you request those. It just takes time. So we go as fast as biology allows us to. We are highly scalable, so we can go from 1 edit all the way to hundreds. We've done for NIH 264 clonal population, and we delivered them in 5 months. That was in iPSCs. That was Alzheimer's studies. We talked about Alzheimer's today, so that scale is also possible, and it's actually very cost effective as well. So we can talk about it if you have any specific projects later on. Just catch me or Hasti later on. But to finish off, I have 30 seconds. I just wanted to leave you with this slide. Maybe you saw it before. It's a meme that is quite popular now on every platform that I saw. But essentially the kitchen and lab are very similar. We follow the protocols, we analyze the results, and the least favorite thing for everybody is cleaning up the mess, right? So I realized that EditCo works similar to Deliveroo, Uber Eats, or however you think about it, right? You can essentially order as many different types of food from one place. They will be delivered to you at the same time. You don't have to worry about the ingredients. You don't have to worry about following the recipe that you have never tried before. So you don't waste probably food while doing that. I mean, I would, because I'm not the best in following the first recipes. But the main thing is cleaning up the mess, right? Like, what if it didn't work? Like, you just, with delivery food, you just chop it away, right? Like, you're ready to throw away basically the packaging that it came in. You can just have a clean kitchen right after it. So think about it as a Deliveroo for CRISPR or cell models, custom cell models. And yeah, that's the end of the presentation. So if you have any feedback, feel free to scan it and leave it to me. If you were counting the DNAs, let me know how many you counted at the break, and I'll give you the coaster. And I'll take any questions if there are any. Thank you.
Thank you very much. Any quick question, or we can also move to the break session and see you at the booth?
Yeah, we don't have a booth.
Oh, you don't
Yeah,
have
we don't have a booth, so just catch me. Like you will see the logo on me and on Hasti. Hasti is